Lipid digestion and absorption requires more steps, and that's because lipids are insoluble in water. Of the lipids in the adult diet, more than 90% or so is consumed in the form of triglycerides. These are also called triacylglycerols. And these molecules are composed of a glycerol head and these three fatty acid tails. And the fatty acid chains make up the hydrophobic portion of the lipid, and the glycerol makes up the hydrophilic portion. A smaller amount of lipids are phospholipids and cholesterol esters, and most phospholipids are glycerophospholipids that are made up of a glycerol backbone linked to two fatty acid tails and one phosphate plus a head group. Triglycerides are digested by lipase, so lingual lipase, gastric lipase, and pancreatic lipase, into monoglycerides and two fatty acids. Phospholipids are digested by pancreatic phospholipase A2 into fatty acids and lysolecithin. And cholesterol esters are digested by pancreatic cholesterol esterase to a cholesterol and a fatty acid. Now let's look at the enzymes that digest lipids and their locations in the GI tract. So lingual lipase digests some lipid in the mouth, but this is a minimal contribution to overall lipid digestion. There's also some contribution by the gastric lipase secreted by the chief cells in the stomach, and the stomach also contributes to the emulsification process by breaking apart large lipid globules into smaller droplets. Then it is the pancreatic enzymes that perform the bulk of digestion in the lumen of the small intestine. Lipase and colipase digest those triglycerides, phospholipase A2 digests phospholipids, and cholesterol esterase, also called nonspecific esterase, digests cholesterol esters. Bile salts from the liver are also important in the emulsification process, and brush border enzymes are not involved in lipid digestion. Lipid digestion and absorption can be divided into 4 stages: emulsification, lipid hydrolysis, also known as lipolysis, micellization, and lipid absorption. So lipids have to be emulsified first because these lipids are insoluble in water. So ingested solid lipids have to be transformed into smaller droplets and eventually into this emulsion of fine lipid droplets so that the water-soluble lipase can come in and digest the lipids. So triglycerides that are consumed in the body are first broken down mechanically through the chewing process in the mouth and the churning movement in the stomach, but these hydrophobic molecules reaggregate in the aqueous environment of the GI tract, forming large lipid globules. The lipase that digests triglycerides is soluble in water, and so it's only able to contact and digest at the surface of the globule. So the solution is to break down the large globule into smaller molecules and then surround them with a stabilizing agent to prevent them from reaggregating. And that's what we see here. There's a large fat globule in the aqueous lumen of the small intestine, and it's broken apart by motility in the stomach and small intestine, and then these bile components come in and surround and stabilize the emulsification droplets. And these are the components of bile. So bile is mostly water, and these are the 2 components of bile that participate in emulsification and, as we'll see later, micellization. Both bile salts and phospholipids are amphipathic molecules, so they have both a hydrophilic region and a hydrophobic region, and that makes it so that they are able to interact with both the lipid and the aqueous environment surrounding it. Other bile components include cholesterol and bile pigments. Now let's look at lipolysis. So triglycerides specifically are digested when lipase comes in and cleaves ester bonds. And that we saw that lipase is present in a few places throughout the GI tract. So lingual lipase is found in the saliva, but only contributes minimally to lipid digestion. Gastric lipase is secreted by the chief cells in the stomach, and it works best in acidic environments. And it cleaves a single fatty acid, so the result from gastric lipase digestion is a 1 fatty acid plus a diglyceride. and gastric lipase is most effective on short to medium chain triglycerides. Pancreatic lipase is secreted by the pancreatic acinar cells in response to cholecystokinin, and it works best in more neutral to alkaline environments. Pancreatic lipase is able to cleave fatty acids off of both sides of this glycerol backbone, so the resulting digestion products are 2 fatty acids plus a monoglyceride. And it's pancreatic lipase that performs the majority of lipid digestion in the GI tract. Pancreatic lipase requires the presence of the protein cofactor colipase. So this is our emulsion droplet with triglycerides, and it's surrounded by these bile salts, and the bile salts prevent the action of lipase. So the pancreas secretes colipase in its inactive form, this procolipase. And then in the small intestine, trypsin cleaves procolipase to produce the active colipase. And colipase is necessary because these emulsifier components at the surface of the lipid droplet displace lipase and prevent it from accessing the surface of the emulsion droplet. So colipase is able to bind both the bile salts as well as the lipase, bringing the lipase into contact with the emulsion droplet and allowing it to digest the triglycerides at the surface. So the result of this lipase digestion is the release of monoglycerides and fatty acids from the emulsion droplets. Then, as these surface triglycerides from the emulsion droplets are digested, they're replaced by triglycerides from the core of the droplet, causing the droplet to become progressively smaller. And as the surface area of the droplet increases, the rate of hydrolysis also increases. Triglyceride breakdown is a highly reversible process, so accumulation of these digestion components in the lumen would block further digestion from occurring. And so bile salts play an important role in removing these digestion components from the vicinity of digestion almost as quickly as they're formed. And this process is called micellization. And this is an example of one of those micelles. We can see the details of what's included in that micelle. So micelles trap and stabilize these lipid digestion products, making them soluble in the aqueous lumen of the intestine. There are 2 additional enzymes of lipolysis, so that phospholipase A2, secreted by the pancreas and responsible for cleaving the fatty acid at that 2 position of glycerol. And it's secreted in an inactive form that is then activated in the lumen of the small intestine by trypsin. Cholesterol esterase is also secreted by the pancreas, and because it's active against a wide range of esterases, it's also known as non-specific esterase. Now that we've digested lipids, let's look at the process of lipid absorption. Out here is the lumen, and we have a micelle present. You can see the different components of the micelle. So the micelle contains the products of lipid digestion, while the exterior is covered with bile salts. So the hydrophilic part of the bile salt is in contact with the aqueous lumen of the small intestine, and the hydrophobic portion of that bile salt is in contact with the lipids. This mixed micelle is then able to transport the digestion components to the brush border of the intestinal epithelial cells where they'll be absorbed. So at the luminal membrane, the lipid digestion components are released from that micelle, and they're able to diffuse into the cell down their concentration gradients. Bile salts from micelles remain in the lumen, and they are taken through the intestine and absorbed in the ileum of the small intestine and taken back to the liver to be reused. Once these digestion components are inside the epithelial cell, the products of lipid digestion are re-esterified with these free fatty acids on the smooth endoplasmic reticulum to form the original ingested cholesterol ester, triglycerides, and phospholipids. The reformed lipids are then packaged with these apoproteins into these large chylomicron molecules. The core of the chylomicron contains the cholesterol ester and triglyceride, which is surrounded by phospholipids and these apoproteins. You can see an example of one of those here with the cholesterol esters and triglycerides at the core and the phospholipids and apolipoproteins surrounding them. Chylomicrons are then exocytosed across the basolateral membrane, and because they're so large, they're unable to enter the capillaries, but instead enter the lymphatic capillaries, where they're carried via the lymphatic circulation to the thoracic duct, which empties into the bloodstream. And this image just shows an overview of all of the steps we've been talking about in one place. So first, these dietary lipids are emulsified into smaller droplets with the aid of the bile salts. This allows lipase and colipase to start lipolysis of triglycerides into monoglycerides and free fatty acids. These digestion components of lipids are quickly taken up inside the micelle, and that's driven by those bile acids. The micelles migrate to the brush border, where lipid digestion components move across the luminal membrane and into the cytoplasm. And then within the cell, digestion components are repackaged, and triglycerides, cholesterol, and those apoproteins combine to create chylomicrons. And these chylomicrons are then released through exocytosis across the basolateral membrane and into the lymphatic system.